Hello everyone and welcome to today's session. We're gonna start in 5 minutes so that more people can join in. Okay? Yeah. hope everyone can see my screen. So we can start our today's class. Apologies for not taking class last Saturday. I was traveling on that day. I'll make sure that I'll take a makeup class for that particular Saturday. Maybe in the weekday, or maybe I can take a 2 hours class in the coming Saturday. Whatever I'm gonna decide, I'll surely inform you well in advance so that you guys can manage your schedule accordingly. Okay, so we will start our today's class. If you remember, in the last class we basically talked about different types of lipids which are present in the membrane, right? So we were generally talking about lipids, though the part which I'm gonna share today first, it's not the part of the curriculum, but I want that you guys should have a basic idea and not only think that the lipids which I'm gonna discuss in the excuse me in the coming class, that these are the only type of lipids which are present in living system. Okay, so we're gonna discuss these lipids which are mentioned here, like PA, PC, PA, PE, PC, PS, PI, PgCl. These are the major phospholipids which are present in membrane. Okay, but apart from them also there are a lot of lipids and types of lipids which are present in a living system. So to give you brief about it, and before going in details about the structure of these particular phospholipids, I would like to make sure that you guys have basic idea about lipids in general. Okay, so for that, let me share my screen with you guys. Yeah, so this is a basic introduction about lipids. So phospholipids are a type of lipids, but these are not the only lipids which are present in the living system. Okay, so just let's go through this video and try to understand and see how diverse the lipids are. Okay,
and oils that you've seen in everyday life. These include fatty acids, triglycerides, phospholipids, steroids, waxes, terpenes, prostaglandins, and things like that. Now, lipids are nonpolar, which means that they're mostly hydrophobic. That is, they don't mix well with water. Hydro means water, phobic means fear, and they try to stay away from water. Now, lipids are mostly made up of carbon-hydrogen bonds. They're hydrocarbons. Now, they may contain other elements like oxygen and sometimes nitrogen in the case of phospholipids, and phosphorus too. But for the most part, they contain mostly CH bonds, and CH bonds are nonpolar. And it's because of the CH bonds that lipids are hydrophobic, that they don't mix well with water. Now, lipids such as triglycerides, they're very useful for long-term energy storage. If you're fasting, if you're not eating for more than a day or so, after your body burns out all the carbs, the next thing that it looks for are the triglycerides in your system, the lipids. And it uses enzymes such as lipase to break down triglycerides into fatty acids and glycerol so that your body can derive energy from these molecules. So they're very useful for long-term energy storage. Your body can go days without food. As long as you have water, you can go many days without food because of these triglyceride molecules. They're very effective in storing energy. In fact, they can store more than twice the amount of energy compared to carbohydrates. Now, lipids also play a major role in the cellular membrane. In fact, the cell membrane is mostly made up of phospholipids. Now, lipids also provide thermal insulation and protection. So they're very useful in the winter when it gets cold. But now let's talk about each of these lipids one at a time. So let's begin with fatty acids. What are fatty acids? Fatty acids are long chains of hydrocarbons that contain a carboxylic acid functional group at the end. So here's an example of a fatty acid. So this right here represents a carbon atom. And those carbon atoms contain hydrogen atoms. Keep in mind, carbon can only form 4 bonds. So the carbon atoms in the middle have 2 hydrogen atoms, whereas the one at the end possess 3 hydrogen atoms. So as you can see, a fatty acid is mostly made up of hydrocarbons. The carboxylic acid portion of a fatty acid is the polar part. So this is the part that likes water. That's the hydrophilic part. And the tail of the fatty acid is the nonpolar part. So that is the hydrophobic part, the part that doesn't like water. Now, this type of fatty acid is known as a saturated fatty acid. It's saturated in that it contains the maximum number of hydrogen atoms that you can pack on this molecule, and there's no double bonds. Saturated fatty acids are solid at room temperature. A good example of this would be butter. An unsaturated fatty acid has a double bond. So it looks something like this. Because of the presence of the double bond, there's a kink in the structure. This molecule, it bends. So that's an example of an unsaturated fatty acid. For every double bond present, you lose 2 hydrogen atoms. Unsaturated fatty acids tend to be liquid at room temperature. So these would include the vegetable oils. Now, another type of unsaturated fatty acid are the trans fatty acid. In this configuration, we have a cis double bond because the hydrogen atoms across the carbons that are double bonded are on the same side of the double bond. In a trans fatty acid, the hydrogen atoms on the double bond are on opposite sides. So that's an example of a trans unsaturated fatty acid. Now let's talk about triglycerides. So what we're going to do is take a 3-carbon molecule called glycerol. Glycerol has 3 hydroxyl groups, and we're going to react it with 3 fatty acids. The first fatty acid is a saturated fatty acid. The second one is a monounsaturated fatty acid because we only have 1 double bond. When you have multiple double bonds, you have a polyunsaturated fatty acid. A triglyceride is composed of 3 fatty acid molecules and 1 glycerol molecule. Now to make it, we need a dehydration synthesis reaction. So we're going to lose water. These molecules will be dehydrated due to the loss of water. So we're going to lose 3 water molecules, and then we're going to synthesize a larger compound. So it's a dehydration synthesis process. And this will produce a triglyceride, which looks like something like this. Now, I might have lost track of the number of carbon atoms I had at the beginning, but I just want to show you the general shape of a triglyceride. So this is an example of a triglyceride. So this molecule is very useful for long-term energy storage. That's one of its major functions. You can pack a lot of energy into this molecule. The triglyceride is nonpolar. It doesn't mix well with water. It doesn't have the polar hydroxyl groups, so it can't hydrogen bond with water. So to review, in order to make a triglyceride, we need to react glycerol with 3 fatty acids, and that's going to make the triglyceride producing 3 water molecules, and that is a dehydration synthesis reaction. But now we can also go backwards if we want to. the reaction between water and the triglyceride can produce glycerol and the 3 fatty acids. Going backwards, the reaction is called a hydrolysis reaction. Hydro means water, lysis means to split apart. So we're splitting apart the triglyceride into its components, glycerol and the fatty acids. Now let's move on to our next category, phospholipids. These are lipids with a phosphate group. So a phospholipid is made up of a phosphate group, a glycerol molecule, and 2 fatty acid chains. In this example, we have a saturated fatty acid and an unsaturated fatty acid. The R group does contain a nitrogen atom, by the way, just in case you have a test question on that. So phospholipids contain carbon, hydrogen, oxygen, phosphorus, and nitrogen. Phospholipids make up the cell membrane. As you can see in this picture, this is the phospholipid bilayer. Now, a phospholipid has a polar head and 2 nonpolar tails. The kink that you see in the structure is due to the presence of this double bond in the unsaturated fatty acid. The phosphate group is polar, so it likes water. So that's the part that's going to be facing water. The 2 fatty acid tails, they don't like water. So this is the hydrophobic region of the cell membrane. That's the part that wants to stay away from water. So this would be the inside of the cell, which is aqueous, it has water in it, and this is outside of the cell. But the membrane of the cell, in the interior, it's hydrophobic. Now the next category of lipids that we're going to talk about are steroids. Steroids contain 4 fused rings, as you can see in the examples listed on the screen. Now the first one that we're going to talk about is cholesterol. Cholesterol is used to maintain the fluidity of the cell membrane. On the upper right, we have estradiol, which is a type of estrogen, the primary female sex hormone. On the bottom left, we have testosterone, which is an androgen, and that is the primary male sex hormone. Estradiol and testosterone, they're chemically similar. Besides both having estradiol and testosterone are chemically similar. Besides both having 4 fused rings, estradiol and testosterone both have this hydroxyl group at the top. But estradiol really stands out due to the presence of this aromatic ring, which is not present in testosterone or in cholesterol. Cortisol is another steroid hormone that is used to reduce inflammation by suppressing the immune system. Now let's say if you're stressed out, you have a lot of exams to study for. When your stress levels are high, your cortisol levels will be high as well. Now when used as a medication, cortisol is known as hydrocortisone. So those are some steroids, which are lipids that you may need to know. Now the next type of lipid that we need to talk about are waxes. Waxes are nonpolar. They don't mix well with water. Waxes contain very long alkyl chains, as you can see in this picture above. But they can have many different functional groups. Here we have a paraffin wax with 31 carbon atoms. The functional group is an alkane. An alkane is an organic molecule that only contains carbon and hydrogen. It doesn't have any double bonds. So it's a saturated compound. Below, we have a wax ester. We still have 2 long alkyl chains, but we do have an ester functional group. So it's called a wax ester. This one has 32 carbon atoms and 64 hydrogen atoms. So these are very, very long hydrophobic molecules. Some examples of waxes include beeswax, I'm sure you heard of that one, or the waxes that plants use to coat themselves in order to regulate evaporation and hydration, and even candles. Candles are made up of waxes, specifically the paraffin wax. Waxes have very high melting points. This particular paraffin wax has a melting point of 99 degrees Fahrenheit, and its boiling point is 698 degrees Fahrenheit. So at room temperature, waxes are solids. But if you raise the temperature, you can melt them into a liquid. Now, the next type of lipid that we need to talk about are terpenes. The basic unit of a terpene is isoprene. Isoprene has 5 carbon atoms, and it's a diene. Whenever you see the suffix "ene," that tells you that it's an alkene. It has a double bond. Di means 2, so a diene is a substance with 2 double bonds. Triene would be 3 double bonds. Other examples of terpenes include myrcene and limonene, which is found in the peels of citrus fruits. And then we have beta-carotene. So this is found in carrots. Myrcene has 10 carbon atoms. So therefore it's made up of 2 isoprene units. So terpenes typically have carbon atoms that are multiples of 5. Now here's a pop quiz for you, or a test question. How many isoprene units are needed to make beta-carotene? What would you say? What you need to do is count the total number of carbon atoms in beta-carotene. So you can pause the video and do that. But you should get 40 carbon atoms. And all you need to do is divide this by 5. This will give you the number of isoprene units that are needed to make beta-carotene. So in this case, that would be 8 isoprene units. If we look at limonene, it has 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 carbon atoms. So 10 divided by 5 will give us 2. So it takes 2 isoprene units to make up limonene. Limonene is an example of a cyclic terpene because it has a ring. So that's it for terpenes, another type of lipids that you may need to be familiar with. Now let's move on to our last category, eicosanoids. So there's 3 categories of eicosanoids that I'm going to talk about: the prostaglandins, the thromboxanes, and the leukotrienes. The prostaglandins have a 5 carbon ring, and they have a total of 20 carbon atoms. The thromboxanes, they have a 6-membered ether-containing ring. The leukotrienes, even though they have 4 double bonds, they have 3 conjugated double bonds. These are double bonds that alternate with single bonds. So these are all lipids. And notice that they all have a carboxylic acid functional group. Prostaglandins are used as vasodilators. They can widen blood vessels. They can also inhibit platelet aggregation. Thromboxanes, they kind of work in reverse. Thromboxanes, they can facilitate platelet aggregation. They have a role in clot formation. And in addition, they work as vasoconstrictors, which means they can make the blood vessel smaller. They can cause it to be more narrow. Leukotrienes are like signal molecules. They serve as inflammatory mediators. So those are some of the functions of these 3 types of eicosanoids. So that's it for this video. That's all I got. Thanks for watching, and if you like it, don't forget to subscribe.
Yeah, so as I've told you earlier, that this video which I've shown you, oh, can you hear me now? Gayatri, you texted audio. I was actually on mute because the video was playing. I hope the audio was available for the YouTube video which I was showing. Yeah, coming back to our lecture slides. So as I've told you, hope you can see my screen also. As I've told you that phospholipids are a type of lipids, and the video which I have shown you, it basically gives you an idea about how different types of lipids are present in the living system, or lipids in general, right? So because we're gonna focus solely on a type of lipid called phospholipid. So you should not get this idea that phospholipids are the only types of lipids which are present in the system. There are multiple lipids. They have different and diverse functionality, as we have discussed in the previous class. Okay, so keeping this thing in mind, if you remember, this was the last slide where we have stopped our discussion in the previous class. This is the generic structure of a phospholipid, which has a glycerol black backbone, and two fatty acids are attached to the glycerol, with one O is being attached with a phosphate group, and that phosphate group have further different alcohols attached to it, based upon which there are n number of phospholipids in the membrane. So this is the general way how we will represent phospholipid. So this will be denoted as the backbone, where these two tails are the acyl chains, this straight line is the glycerol, and this is the phosphate group which is attached, and this alcohol will be the will be different groups for different types of lipids. So we will discuss maybe 2 to 3 different types of lipids for today, and remaining we will cover in our Saturday class. Again, I am mentioning, depending upon the schedule, I might take a 2-hour class on Saturday for making up my class which I have missed last Saturday, but I am not certain. I will certainly share the information before the class, or I might schedule the makeup class in the weekday itself, but the chances are I might take 2 hours class in the on the coming Saturday. Okay, keeping this in mind, we gonna go to our first type of lipid, which is called phosphatidic acid. So phosphatidic acid, or PA in short. As you can see, the representation of phosphatidic acid is again the glycerol backbone and the acyl chains and the phosphate head group, and in place of alcohol it has simple H attached to it, right? Simple H. So some basic characteristics of phosphatidic acid is, phosphatidic acid is called PA in short, and phosphatidic acid, PA, is the smallest and simplest phospholipid, and so is the precursor for other more complex alcohol containing phospholipids, right? As you can see, the alcohol which is attached to the O group of the general representation of the phospholipid is literally a single OH group, hence it is the simplest, right? Because the simplest molecule you can attach to the O is the H, correct? So this is the simplest phospholipid which is present in the membrane. As it is the simplest, which means it can act as a base for generating and giving rise to other type of phospholipids. Okay, so at physiological pH, PA has a net charge of about -15. I apologize for this question. It is minus indicating that this particular lipid is negatively charged. Okay, and so is a strong anion that contributes negative charge density to the membrane. PA has a unique shape, possessing a small, highly charged head group whose charge lies very closely to the glycerol backbone. So what it says, that why it is so special and unique in shape, because see, here is a negative charge, right? And this, you know that this comprises the head group and this comprises the tail, right? So head group, as it doesn't have any extended alcohol groups attached to it, the negative charge lies very closely, it is not dispersed. This negative charge lies very closely to the tail, correct, indicating that a lot of charge is saturated on a small head and the tails are closer to the head. Okay, so PA's shape allows this phospholipid to induce high membrane curvature stress and is probably related to its significant role in membrane vesicle fission and fusion events. Now you need to start looking at these lipids with respect to the functionality of the membrane, right? As I told you earlier, that the distribution of lipids, the components remain same, and composition varies depending upon the function in which that particular membrane or cell or the part of membrane which you are talking about is responsible for. Okay, so it has been observed that because phosphatidic acid, that is PA, is a simplest phospholipid, it has concentrated negative charge, it is found that wherever phosphatidic acid or PA is present, it is responsible for high membrane curvature stress, and most likely it might be also associated with vesicle fission, because it's a small molecule, right? It will induce curvature to the membrane, and hence anything which needs to be detached from a membrane which has less curvature, it needs to induce a lot of curvature, right, to the membrane. We might see some things related to it in future, how this blebbing of membrane, like if you remember apoptosis and necrosis, all these have like membrane disrupting qualities, that they turn into small droplets, which again macrophages comes and clean them, right? So these small droplets also, there has to be a curvature inducing molecules present there to ensure that the excuse me, that the membrane is formed, and hence there phosphatidic acid plays a significant role. Okay, PA's shape allows this phospholipid to induce high membrane curvature stress and is probably related to its significant role in vesicle fission and fusion events, right? Both fission and fusion will require change in the curvature and induction of the curvature, correct? Therefore, wherever there is dynamicity associated with the membrane, via which the membrane needs to either fission out, like make vesicles, or fusion in, which means something is coming to the membrane and fusing in, chances are PA will be at the interface and inducing a curvature to the membrane. PA is a vital cell lipid that is maintained at extremely low levels in the cell by the activity of potent lipid phosphate phosphohydrolases. Lipid phosphate phosphohydrolases rapidly convert PA to diacylglycerol, a metabolic precursor of many lipids. As I have mentioned, that PA is the simplest form of phospholipid, it will act as a precursor for other lipids. So PA is generally not maintained at a very high level in the cell. It is generally converted into DAG, and this DAG is further responsible for generating multiple types of lipids. We're gonna see maybe today or in the next class, depending upon our time, that few of the lipids which I'm going to discuss today are interrelated, meaning that their alcohol group is somewhat interconvertible, and this plays a huge role in maintaining their levels in the in our cell. Okay, so this is the basic information about phosphatidic acid. Now if we talk about the next lipid in our list, it is the phosphatidyl ethanolamine, that is PE. You can see the representation of PE here. The alcohol group which comprises the ethanolamine is CH2, CH2 and NH3, correct? And you can see the net charge on PE will be, depending upon the physiological pH, we can say that it will be either neutral or it might have slightly negative charge. Okay, so if we talk about phosphatidyl ethanolamine, that is PE in short, is infrequently referred to as cephalin from the word cephalic, meaning pertaining to the head. It is the second most prevalent phospholipid in humans, but is the principal phospholipid in bacteria. In us or in humans, PC is the most abundant phospholipid, while PE comes later. But in bacteria, PE is the dominant phospholipid. Why? Because humans have the ability to convert PE to PC, right? As I have mentioned, there are 3 to 4 lipids which are interconvertible to each other, and this ensures that their levels are properly maintained in the cell. So humans, we have the ability, we have the enzymes which can convert PC to PE or PE to PC, right? Whereas bacteria cannot do this. So bacteria are relegated to having PE as their major membrane phospholipid. Okay. In humans, PE is found particularly in nervous tissue, including the white matter of the brain, nerves, and in the spinal cord. Because the pKa of the PE amine is about 8.5, the pKa of this is 8.5 at physiological pH. The amine is mostly but not completely protonated, okay, with the fully dissociated phosphate. PE have a slightly negative net charge, okay. The next lipid which we going to talk about today, these are very generic description about these lipids, because I want you guys to know what are the different types of phospholipids present and how they vary from each other. So if we talk about phosphatidylcholine, that is PC in short, you can see the alcoholic group which is attached to the phosphatidylcholine, or the choline group, is comprising basically of CH2, CH2N, and all the H of NH3 being replaced with CH3. Okay, so PC is the major phospholipid found in animal membranes, and therefore is the most abundant membrane structural lipid component. The molecule is can shaped, with its polar head groups being about the same width as the sum of its apolar tails. We're gonna see different shape of these lipids, and then you will get more idea about how and where these lipids might be present in the membrane. Not in today's class, but definitely in the Saturday's class we're gonna see how these lipids can be represented based upon the shape. Like some lipids are can shaped, as you can see here, it's like PC is can shaped, then there will be a cone shape, then there will be inverted cone shape. Okay, so these are some basic shapes which are associated with certain type of phospholipids, which allow us to get an idea about the curvature which they gonna induce in the membrane. Okay, in addition, PC is a true Zwitter ion possessing a formal positive and negative charge but no net charge. Okay, therefore PC does not exhibit negative charge repulsion, a potential problem with most other phospholipids. Okay, talking about phosphatidylserine, that is PS. So this is bit more complex, but still you can see that this is the phosphate group and the alcohol group is CH2 CH, and with CH there is a amine group, and via amine group then there is a carboxylic group. Okay, then the major function of PS or phosphatidylserine is associated with the brain, where it is far more abundant than in other organs. PS is involved in both activating and anchoring a variety of proteins to membranes. Included in this long list of proteins are several ATPases, kinases, receptors, and the crucial signaling transducing proteins, protein kinase C, and adenylated cyclase. Correct? So PS is a very different and unique type of phospholipid. It is majorly present in membrane. It actually helps in anchoring and activating certain types of lipids. So there are certain lipids which are actually important for the structural integrity of the protein. So we're not gonna go in too much detail about it, but you guys should know that if the membrane is there and proteins are associated with the membrane, it is not only the proteins which are working, right? Because they are in so close proximity, there is a dependence of some type of lipids in the functionality of the protein also. Okay, normally PS is located almost exclusively in the membrane inner leaflet. Okay, as you can see, it might have a net negative charge or net positive charge. This question is on you guys. You have to tell me what will be the net charge on phosphatidylserine depending upon the structure which you can see here at physiological pH, obviously. Okay, So till now we have talked about four lipids, sorry, right? Phosphatidylserine, phosphatidylcholine, phosphatidylethanolamine, and phosphatidic acid. So these four excuse me. Sorry for that. So these four lipids, that is PA, PC, PS, and PE. These four lipids, as you can see in their backbone, they are very similar. I'm gonna show you maybe in the next slide if I have no. In the next class, I'll show you how you can actually interconvert these four types of phospholipids. So if we talk about the composition, right, as I've mentioned that there are certain lipids which are more abundant in bacterial membrane. There are other phospholipids which are more abundant in mammalian cells. So if we look at the average composition of a mammalian liver cell, we can clearly see that the mole percentage of PC is among the highest, that is more than 55, or say definitely more than 50% of the mammalian liver cell has PC, followed by PE. Then we haven't discussed about PI, it will be PI, then PS, PA, CL, SM, and cholesterol. Don't worry about these small terms, we have actually kind of discussed. For example, PI will be phosphatidylinositol, PG will be phosphatidylglycerol, PCL will be cardiolipin, and etc. Okay, so this is a very generic composition of a mammalian liver cell. You need to know this, that this composition again will vary within an organism depending upon the organs from which we are considering the membrane, right? So if you look at the broad distribution of components, especially lipids, different types of lipids in different membrane system, correct? So we can see that the plasma membrane has more cholesterol, correct, while there is negligible cholesterol in the inner mitochondrial membrane, correct? If we look at the plasma membrane, phosphatidylcholine, PC, right, this is present in okayish amount, or while PC of phosphatidylcholine is more abundant in the inner mitochondrial membrane. If we talk about cardiolipins are completely absent in plasma membrane as well as in the nuclear membrane, as well as rough ER and also Golgi, correct? While cardiolipins are comprising among like top 3 most abundant lipids in the inner mitochondrial membrane, while a remnant of cardiolipin is also present in the outer mitochondrial membrane, correct? So if we look at other lipids also, if we look at, for example, let's talk about sphingolipids. So sphingolipids are present in all of the membranes, but to different percentages. Highest will be in lysosome. Sphingolipids are majorly present in lysosome in comparison to the other membranes which are present here, correct? So this actually indicates that the composition of lipids play a vital role in determining what sort of functions the cells are involved in. Okay, the functional specialization of each membrane type is reflected in its unique lipid composition. Cholesterol is prominent in plasma membrane but barely detectable in mitochondrial membranes. Cardiolipin is a major component of inner mitochondrial membrane but not of the plasma membrane, correct? Phosphatidylserine, phosphatidylinositol, and phosphatidylglycerol are relatively minor components of most membrane but serve critical functions. Phosphatidylinositol and its derivative, for example, are important in signal transductions triggered by hormones. Sphingolipids, phosphatidylcholine, and phosphatidylethanolamine are present in most membranes but in varying proportions. Excuse me. Glycolipids, which are major components of the chloroplast membranes of plants, are virtually absent in animal cells. cell. Okay, so we can actually see how different phospholipids are present within an organism, in different, not even an organism, within a cell. They vary among the within the membranes of an organelle, plasma membrane of a single cell. So this indicates how diverse the function a cell can perform just by varying its lipid composition. So this is all for today from my side. We're gonna discuss about the remaining structures of the lipids. Today we have discussed about PA, PE, PC and PS. In next session we're gonna discuss about PI, PG and Cl, and then we're gonna go towards maybe proteins, and before that we're gonna discuss about some properties of the lipids such as excuse me, fluidity, melting point, membrane curvature, bending, and all those physical aspect of a membrane. Okay, feel free to approach me if you guys have any question, and I'll be happy to answer.